Del Monte Food Inc. created the patent for a pineapple plant named Rosé. This is a new variety of pineapple known as Ananas commosus, a fruit that presents pink instead of yellow flesh. Creation process started in 2005, the patent was accepted in 2015 and received FDA approval in 2016, but wasn't available on the market until 2020.
This product has different uses. Like most transgenic plants, it has been designed to have pest and disease resistance. One of its key factors is its delayed ripening. Additionally, they contribute to improve the quality by addressing fruit browning concerns. Also, the pink flesh pineapple stands out for its added health benefits. With its high lycopene content, this pineapple variety has been associated with a reduced risk of specific cancers and heart disease.
Del Monte also patented the transformation method, which involved the cultivation of organogenic pineapple cells with 18 efficients, which is a technique for the propagation of pineapples using vegetable tissues, and 18 efficients is a gram-negative bacterium that causes the disease known as gall in plants. This bacterium is capable of transferring its genetic material to plant cells. RNA interference constructs that were designed to suppress endogenous expression of each lycopene beta cyclase and lycopene epsilon cyclase, and RNA interference construct was inserted, which is intended to suppress endogenous expression of 1-aminocyclopropene 1-carboxylate acid synthase. The penultimate step is ethylene biosynthesis, which induces the flowering in pineapples. Additionally, Nicotiana tabacum acetohydroxy acid synthase cassettes were also added to act as a marker for sulfonylurea selection during transformation.
The pink glow gets its unique color from the expression of the PSY gene found in tangerines, also known as Citrus reticulata.
The genetically modified organism was Del Monte Gold pineapple, also known as MD2. The genetic modification it possesses modulates the carotenoid pathway that gives it the yellow color by producing B-carotene protein. The difference is pink glow expresses the tangerine PSY gene, which limits enzymes during this biosynthesis, producing that pink color. The transformation of MD2 pineapple cells was done by Agrobacterium tumefacius mediated transformation of organogenic tissues. These bacteria contained either a plasmid for increased carotenoid biosynthesis or decreased ethylene biosynthesis. The medium used contained chlorsulfurum that allowed for the selection of positive pineapple transformants. Once pineapple had grown in tissue culture, propagules were taken to a research facility located in Costa Rica. Propagation in field and laboratory were evaluated. Growth rate and characteristics are the same as MD2 except for the color.
Dengue is an acute infection disease of viral etiology. It's a vector-borne disease caused by a positive stranded RNA virus of the family Favibridae. Dengue virus is transmitted to humans by the mosquito Aedes aegypti. More than half of the world's population is at risk of dengue infection. Each year, an estimated of 390 million dengue infections occur around the world, resulting in up to 36,000 deaths. This is why, to prevent the disease outbreaks, the US-owned company Act6 decided to develop a patent for the production of genetically modified AI mosquitoes for the environmental use. Act6 genetically modified Aedes aegypti mosquitoes are mass-produced in a laboratory to carry two types of genes: a self-limiting gene that prevents female mosquito offspring from surviving to adulthood, and a fluorescent marker gene that glows under the special red light. This allows the researchers to identify the genetically modified mosquitoes in the wild.
The self-limiting gene produces the protein TTAB, a protein that binds to its own control regions to set up a positive feedback loop. This loop produces more TTAB, which, in overproduction, prevents the expression of the insect's essential genes, resulting in the shutdown of normal gene expressions that leads to death. During production, tetracycline is added throughout the diet to block the TTAB from binding transcription machinery. This allows insects to survive, in order for large-scale production to be available.
A sterile insect technique, or SIT, involves the imposition of genetic load-dominant lethal mutation by mass reading, sterilization by chemical or radiation, and lastly, the release of large numbers of males to mate with wild.
To control the modified mosquitoes and to differentiate them from the modified ones, the genetically modified mosquitoes were microinjected with a plasmid containing the 5C actin promoter and the marker GFP gen from the jellyfish Aquaria victoria, producing a fluorescent protein called DS-RED2. This protein is found throughout the body of the larvae, pupae, and adults, and glows under a special light, making the offspring easily identifiable. The self-limiting gene can be adjusted to function only in females, to enable large-scale and simple production of the genetically modified mosquitoes, which the company calls the friendly males. The friendly males are released into the wild, and they mate with the female pests. The female offspring do not survive, resulting in targeted decline of the pest population. The mosquito Aedes aegypti has a strong preference for biting people and adapts well to living in urban areas. The EPA evaluated the potential risk of releasing genetically modified mosquitoes into communities and determined that there is no risk to people, animals, or the environment, making it a precise and sustainable solution to the dengue problem, with the capacity of preventing millions of infections that compromise the life quality of society as a whole.